Hey there, and welcome to This Explainer. Today, we're going on a journey completely unlike any other. We're leaving the big, visible world behind and setting the stage for an incredibly exciting microscopic safari adventure right into the hidden, super busy world of cells. Whether you're a seasoned science enthusiast or a curious kid just starting to explore, grab your gear and buckle up, because what we're about to see is absolutely fantastic. Seriously, just imagine for a second that we could shrink ourselves all the way down. I'm talking way past the size of an ant, smaller than a tiny grain of sand, zooming right into the very building blocks of life itself. What would we actually see? Well, we wouldn't find a bunch of empty, quiet space, that's for sure. We'd find bustling, wildly complex microscopic environments that make up every single living thing on Earth. And yeah, that includes you and me. All right, Section 1. Welcome to Cell Safari, the fundamental units of plants and animals. Okay, let's dive right into this. Get ready to explore this hyperactive microscopic world. You know, when we look at a leaf on a tree or even the skin on the back of our hands, we just see a single solid surface. But underneath all of that, there is a highly organized metropolis functioning 24/7. These environments are alive, they're super dynamic, and they're doing all the heavy lifting to keep plants growing and animals moving. Moving on to Section 2, Animal versus Plant Cells. Time to meet our safari subjects. Here are the two main characters of our safari. On the left, check out the typical animal cell. Notice its shape? It's kind of a flexible, irregular blob, right? It doesn't have a rigid outer wall, which is exactly why animals can move, stretch, and dance around. Now, take a look at the typical plant cell on the right. It looks way more like a structured brick, bounded by a thick outer layer. They're both cells, but visually, they have completely different architectural styles to suit their totally different ways of life. But hey, the differences go way beyond just how they look. Animal cells, yep, the exact ones powering your body right now, they store their energy, their carbohydrates as glycogen. They also have nervous coordination, meaning they can communicate incredibly fast to respond to the environment, and they allow organisms to actually move from place to place. Plant cells, on the other hand, well, they play a completely different game. They store their carbs as starch or sucrose. They don't have nervous coordination. Instead, they make their own food through photosynthesis. It's a huge, fundamental divide in the microscopic animal and plant kingdoms. Section 3. Tour of the organelles. Let's look at the inner workings of the cell. Okay, it's time to venture deeper into the cell city to check out the specialized structures that actually do the work, called organelles. First up, the mighty mitochondria. These are literally the cell's power plants. This is the site of aerobic respiration, which is basically where energy is released to power the cell. If a cell has a really active, demanding job, like the muscle cells in your legs when you're running around, you're going to find these tiny powerhouses in extremely high numbers. They're the engine of our microscopic safari. Next on our tour, we have the ribosomes. If the mitochondria are the power plants, ribosomes are the microscopic manufacturing factories. They're these tiny structures that act as the main site for protein synthesis. Basically, they diligently manufacture the proteins needed to keep the organism running. You can find them floating free or attached to a membrane system called the endoplasmic reticulum. When they are attached, it looks all bumpy under a microscope, which gives it fantastic, very scientific-sounding name, the rough endoplasmic reticulum. So here's a really cool point. Both our animal and plant cell friends share a core set of essential organelles. They both have a nucleus, which is basically the boss, containing the DNA genetic material that controls everything. They both have cytoplasm, this gel-like substance where all the chemical reactions happen. They both have a cell membrane, holding it all together and acting like a strict security guard to control exactly what enters and exits. And as we just learned, they both totally rely on ribosomes for proteins and mitochondria for energy. But remember that rigid, brick-like shape of the plant cell we saw earlier? Let's talk about the exclusive features found only in plant territory. Plant cells have a tough, rigid cell wall made of cellulose for extra support. They also have chloroplasts, which contain those green chlorophyll pigments to absorb light energy. Yep, this is exactly how they do photosynthesis. And finally, they feature a large permanent vacuole filled with cell sap, which is a solution of sugars and ions used for storage and to keep the cells structurally supported from the inside out. It's like an internal water balloon, keeping everything firm. Which brings us to Section 4, the tools of discovery, how we capture all this microscopic action. So now that we know what's actually down there, how do we capture this action? what does our safari camera equipment actually look like? Well, scientists primarily use two main tools, and they offer wildly different views. The light microscope directs light through a really thin sample and focuses it through lenses. It's a total classic, great for revealing larger structures like the nucleus, the cell membrane, and those green chloroplasts. But if you really want to zoom in, you absolutely need an electron microscope. This offers vastly higher magnification, revealing tiny, intricate organelles that standard lights simply can't catch, like little transport vesicles and those tiny protein-making ribosomes. Seriously, when we upgrade from the standard light microscope on the left to the incredible electron microscope on the right, it is literally like putting on a pair of prescription glasses for the very first time. Suddenly, bam, a whole new world of tiny, complex structures just pops out at you. The rough endoplasmic reticulum becomes clearly visible, the little inner folds of the mitochondria appear. It takes our understanding from a blurry, basic outline to a stunning high-definition map. Of course, to see anything at all, we've got to prepare our specimens super carefully on a little glass slide. Let's walk through it together. Imagine you're a real lab scientist for a second. For a liquid specimen, you add a few drops of your sample using a pipette. Then, you place a cover slip right over it. The trick here is to gently press down to remove any air bubbles. Trust me, bubbles look like giant, confusing alien rings under a microscope. And, super importantly, always wear gloves to make sure you aren't accidentally looking at your own skin cells instead of your specimen. Now, solid specimens, they require just a bit more finesse. A classic safari subject here is a piece of onion. You have to peel or cut a very, very thin layer of cells, so thin that the light can actually pass right through it. Sometimes you have to treat it with chemicals, and again, you gotta press out those pesky air bubbles with a cover slip. But the real secret weapon here is applying a stain. See, a lot of cell parts are basically transparent. A drop of stain acts like pure magic, suddenly making those invisible organelles pop out in vibrant color directly to your eye. Section 5, the art of cell drawing, sketching the wildlife. All right, you've prepared your slide, you've looked through the microscope, and you've found your specimen. So how do you properly record the wildlife we've just discovered? Well, I need you to channel your inner, incredibly strict art teacher right now, because biological drawing is absolutely not about creative expression. It's all about precise scientific record keeping. And yes, there is a very strict rulebook. You must include a title and magnification level. You've got to use a sharp HB pencil on plain white paper. Your lines must be clear and single, no sketchy, feathery, artistic strokes. And here is the big one. Absolutely no shading allowed. None. You must ensure proper proportions, draw large enough to actually use the space, and your label lines, they must be drawn with a ruler, perfectly parallel to the top of the page, and they must never, ever cross each other.
Let's take a look at a picture-perfect example. Here's an animal cell viewed under an electron microscope, alongside its corresponding biological drawing. Notice how ridiculously crisp it is? The unshaded single lines perfectly follow the rulebook. It identifies the mitochondria, the nucleus, the vesicles, all without a single smudge of artistic shading to confuse what is a real structure versus what is just a shadow.
And right here, we have a brilliant plant cell drawing. See how elegantly those straight, parallel label lines identify all the parts? The cell wall, the chloroplasts, and that massive permanent vacuole are all pointed out without creating a tangled, messy web of arrows. It is a perfect, purely scientific record of exactly what was observed through the lens. Okay, if there's one golden rule of scientific observation you take away from this explainer, it's summed up in this fantastic tip from the examiners. When producing a biological drawing, it is vital that you only ever draw what you see and not what you think you see. It is the ultimate golden rule. Your brain might know that mitochondria are supposed to be there, but hey, if your microscope doesn't show them, your pencil shouldn't draw them. And wow, that brings us to the end of our explainer today. We've shrunk down, we've explored the incredibly busy microscopic cities of plant and animal cells, we've seen the awesome power of electron microscopes, and we've learned the remarkably strict art of recording our findings. So now that you have your official safari training, I want to leave you with this question. What biological specimen would you want to look at under a microscope? Keep that curiosity alive, keep asking questions, and